Picture your DNA as a priceless cookbook, filled with every recipe your body could ever need to function, grow, and survive. This cookbook is locked away in the cell's nucleus for safekeeping, protected from the hustle and bustle of the rest of the cell. When your cell needs to make a specific protein, it doesn't risk damaging the original DNA. Instead, just a single recipe is carefully copied, ensuring the master copy stays untouched. This copy, much like jotting down a recipe on an index card, leaves the safety of the nucleus and travels to the cell's protein factories, known as ribosomes. The process of making this working copy is called transcription, where the DNA's instructions are transcribed into a new language the cell can use. This is how your cell protects its precious DNA, keeping it safe behind a protective barrier, while still getting all the work done that keeps you alive. The cell can make as many recipe copies as needed, sending out multiple messengers at once, all while the original DNA remains secure and undisturbed inside the nucleus. This messenger copy is called mRNA, short for messenger RNA. It's the vital link between your DNA and the proteins that build and run your body. Without mRNA, the DNA's instructions would stay locked away, never reaching the parts of the cell that need them, and your body couldn't function. Transcription unlocks your genetic code, turning stored information into real action, making the proteins that keep you alive, healthy, and uniquely you. Let's meet the stars of transcription. First, the DNA template strand, one half of the double helix holding the recipe to copy. Next, RNA polymerase, the enzyme that reads the DNA and builds the RNA message. It unzips the DNA, reads each letter, and assembles a matching RNA strand. The result? Messenger RNA or mRNA, a single-stranded flexible copy ready to leave the nucleus. Unlike DNA, mRNA uses uracil, U, instead of thymine, T. These three, DNA template, RNA polymerase, and mRNA, are the core team. Each plays a vital role in getting genetic instructions out of the nucleus. Without any one of them, protein production would grind to a halt. Transcription unfolds in 3 acts: initiation, elongation, and termination. These steps are essential for turning the genetic code stored in DNA into a working copy that the cell can use. Each phase is carefully orchestrated, ensuring the right information is captured and transferred. It all starts at a special region called the promoter, a start here sign on the DNA. The promoter acts as a landing pad, signaling where transcription should begin and helping the cell know exactly which gene to copy. Here, the enzyme RNA polymerase binds tightly to the promoter, unwinds the DNA double helix, and exposes the template strand. This template will guide the creation of a new RNA molecule. During elongation, RNA polymerase travels along the DNA, reading its code and adding matching RNA bases one by one. This process builds a growing mRNA strand, which is a mirror image of the gene's instructions. Instead of thymine, RNA uses uracil, U. So for every adenine, A, it reads on the DNA, it adds a uracil to the mRNA. This small change is a key difference between DNA and RNA. As the mRNA strand grows, it peels away from the DNA, allowing the DNA to zip back up into its original double helix form. This keeps the genetic code safe and ready for future use. Termination happens when RNA polymerase reaches the end of the gene. A stop signal in the DNA tells the enzyme to let go, releasing the finished mRNA strand. The DNA rewinds itself, and the new mRNA is now complete and ready for the next stage in gene expression. In just these 3 steps, the cell creates a portable working copy of a gene, an mRNA molecule that can leave the nucleus and guide protein production. This entire process is remarkably fast, incredibly precise, and tightly controlled to ensure the cell's survival and proper function. But before mRNA can fulfill its role, it must undergo some important editing and processing steps to become fully functional. The first mRNA copy, pre-mRNA, is a rough draft. It needs editing before leaving the nucleus. First, a protective cap is added to the front, helping the ribosome find the message and shielding it from damage. Next, a poly-A tail is added to the end, boosting stability and export. The most dramatic edit? Splicing. Introns, non-coding sections, are snipped out, leaving only exons, the useful instructions. Specialized machines called spliceosomes handle this cut-and-paste job. Once capped, tailed, and spliced, the mature mRNA is ready. It exits the nucleus through a pore, heading for the ribosome. Now it's set to deliver its instructions for building a protein. Why does transcription matter? Why should we care about this process that happens deep inside our cells, invisible to the naked eye? It's the bridge between DNA and life itself, a vital link that transforms genetic instructions into action. DNA holds the code for life, but without transcription, it's just a silent archive, an instruction manual that never gets read. Through transcription, genes are copied into RNA, and from there, the magic happens. Proteins, built from transcribed genes, do everything. They speed up chemical reactions, build the structures of our bodies, transport oxygen in our blood, and fight off disease. Every response, from making insulin to regulating metabolism or defending against germs, starts with transcription. It lets cells turn genes on or off as needed, responding to signals from the environment, stress, or development. This is how each cell type becomes unique, how a skin cell knows to be skin, and a neuron knows to be a neuron. Transcription is the cell's main control point, the master switchboard for gene activity. This process is managed by specialized proteins called transcription factors, which decide which genes are turned on or off at any moment. Mistakes here can cause disease, from cancer to genetic disorders, so precision is absolutely vital. Understanding transcription is key to breakthroughs in medicine and genetics, helping us diagnose, treat, and even prevent disease. It's the first step in the flow of genetic information, a process known as the central dogma of molecular biology. DNA makes RNA, and RNA makes protein. This is the fundamental pathway that powers all living things. Every heartbeat, every thought, every breath you take depends on this elegant, microscopic process. Transcription, the true engine of life. Let's recap the journey. DNA stays safe in the nucleus. The cell copies just what it needs through transcription. The team DNA template, RNA polymerase, and mRNA. The steps: initiation, elongation, termination. The mRNA draft is edited, capped, tailed, and spliced, then sent out of the nucleus. This process links DNA to protein, powering nearly every function in your body. But the story isn't over. Next, mRNA meets the ribosome for translation. That's a tale for another day. For now, marvel at transcription, the cell's way of turning genetic code into life.